right after where the schedule is, the table that has a layout of all of the the class meeting times and all of that. The next thing in there, resources or something like that. There we go. Just scrolling down. So there's that table that has the schedule in it. And then after that, it says online resources. This is where you would go to, maybe previously have gone to get to the Google Drive location where I have the audio recordings of old lectures. Now I'm switching to doing it this way, so you can get to that through this link that says YouTube channel. Also, what I'm using, the presentation software that I was using last time, and I'm going to continue with. You can go to see those through the Prezi account page. Now, Janelida, you said that you had an issue with that once. That
Which one?
you clicked here and it took you to something asking you to log in.
Mm-hmm.
Has anybody else used this link?
It worked fine for me.
Okay. So I have a link up here that says Prezi login for me to get into my account to do changes. That link should be different from this link. So I don't quite know what was happening with you. Try it again.
If that happens to you, you can go to the Prezi homepage and you can search users, and I'm assuming your username is just your name.
Right, should be.
Just put in his name.
So you
and
should
it would
find it
show up pods that he made.
But if you try it again, you still have that problem, even just straight clicking on that, let me know, because there's something weird about that.
Okay.
So yeah, that's where you can get to it. When you see my cute little face there, you know you've gone too far. So, okay. So last time we got to talking about the structure of the red blood cell. I kind of ended quickly by talking about hemoglobin. I don't remember if I mentioned it or how explicit I was about it, but I was showing that hemoglobin has 4 subunits. There's the globin subunits. I might not have gotten to the next thing in this talk specifically, but sickle cell. Yeah, I did show you that picture of the sickle cell. Yeah, so that's where we ended. And then from there, I went on to this picture, just say, okay, this is where we're going to start up. It would have been nice to have wrapped up all of the red blood cell stuff in the first day, but there wasn't quite time for it. So the life cycle of the red blood cell is kind of unusual. It's not what you tend to see for any kind of cell. They have a limited lifespan. They last for about 120 days, about 4 months. The reason for that essentially comes down to they're exposed to one of the most toxic molecules that's in your body right now. That's oxygen. Okay. A lot of people kind of don't realize that. You think of oxygen as being very important for what goes on in your body, but actually it's a potentially very toxic molecule. The reason for that is that it's a reactive molecule, or the oxygen atom is a very reactive atom. A lot of things can be oxidized. And some of the theories about aging and cell deterioration are oxidative stress theories. You might have heard that. Or more likely, you've heard of antioxidants. You might have heard of superfoods that are chock full of antioxidants, or taking some sort of vitamin supplement that'll have antioxidants in it. Antioxidants are chemicals that react with oxygen so that oxygen doesn't accidentally react with molecules in your body. This theory of aging is that things like DNA get oxidized over the years that we're alive, and eventually the damage from those oxidative reactions adds up and our cells stop functioning. Now, that is not a very comprehensive theory on what's going on behind aging. If it were true, then antioxidants would make us live forever, and that's not true. But there are some suggestions about how controlling oxidative stress can actually help some cells function better. But red blood cells, since their job is to carry oxygen, are basically on the front lines of oxidative stress. So we don't want them to last our entire lives because they'll accumulate a lot of oxidative reactions, and that can cause lots of problems. So they have this limited lifespan. They're made in the red the red bone marrow, and then they go out into circulation where they circulate for about 120 days. And I got to get in the habit of using the mouse to point at things for people that are watching this. So they start in the red bone marrow. They last for about 120 days. And then they are engulfed by a type of white blood cell called a macrophage. The word macrophage means big eater, kind of. These are cells that go around and basically gobble up all of the pathogens and debris and whatever there is in your body. And one of the things they do is they gobble up old red blood cells. They then break the old red blood cells down. The cell membrane's taken apart. The proteins inside it are taken apart. The other components of it are broken down, mostly for recycling. So the cell membrane components are recycled to be used in other cell membranes elsewhere. The amino acids that are removed by breaking down proteins like hemoglobin are recycled to be used to make new proteins elsewhere. The iron that's found within the heme molecule is removed, and it's specifically transported back into the red bone marrow to be incorporated into new hemoglobin molecules. There's a molecule called transferrin, which is responsible for doing that. Now, the word transferrin looks like it has transfer in it right there. But in fact, the fer part of transferrin comes from the Latin word for iron, which is ferrous, as in a Ferris wheel. So transferrin is what carries iron around in your body. And so what's left over from all of that is the heme molecule, which is what was in the hemoglobin molecule that actually held onto the iron. Instead of being broken down and recycled to make more heme molecules or whatever, it gets repurposed. It's changed into an intermediate molecule called biliverdin, and then into a molecule called bilirubin. Now, the bil in both of these words refers to bile, which is a digestive juice. It's made in the liver. The liver secretes bile into the small intestine, which is necessary to help us break down lipids. And when we get to the digestive system, I'll talk about that process a bit more. Bile doesn't enzymatically break down lipids itself. Instead, it gets in and wedges itself between lipid molecules, which are hydrophobic and therefore would like to stick together. So it breaks them apart from each other so get in and break them down. And bilirubin is one of the molecules that does that it can kind of get in between lipid molecules so they can be pulled apart. In doing that, it's not changed at all, okay? It doesn't go through any chemical reactions to do that. It just mixes in and separates things out a bit. Some of that, which is not actually depicted in this picture here, gets taken back up out of the small intestine and sent back to the liver to continue being part of bile. But some of it gets into the small intestine. It doesn't get taken back up into the bloodstream. So it stays in the small intestine, moves through the digestive system, and eventually gets to the large intestine, where it gets changed into a couple of other types of molecules. One of them is called urobilinogen, which belongs to the group that's named there, the urobilins. And another one is stercobilin. Now, the uro in urobilins or urobilinogen refers to urine, although some of that doesn't go into the urine. Here it says absorbed into the circulation, and then they go to the kidneys to be excreted in urine. That's a little bit oversimplified because a lot of those never make it in circulation and they stay in the digestive tract. But stercobilin just stays in the digestive tract. Now, a lot of these molecules that heme molecule gets made into are actually pigment molecules. And in their name, they actually refer to the color pigment that they are to some degree. Verdin in biliverdin means green. I assume it's green. I actually have no idea because it's this intermediate molecule. It doesn't do a whole lot other than gets made into bilirubin. Rubin in that word comes from the same place we get the word ruby, means red. Unusually though, bile is yellow. So I don't know why this is a red pigment molecule and it ends up being, bile ends up being yellow. I'm not a chemist, I don't know why that happens. But, and then stercobilin, the color isn't there, but isn't in the name of the molecule, but it is a colored molecule. And its claim to fame is that it's what gives feces their color. So it's gives the tannish to brown color to feces. When the color of your poop doesn't match what you expect it might supposed to be, it might be something to do with this. It might be a liver function problem. It might be something with heme way back at the beginning, whatever. So it's in there. Again, when we get to the digestive system, we'll talk more about the liver and bile and what's going on in the intestines. But what we're seeing here is the pathway that this molecule takes as it goes through all of this. So, just to get back to this for a second. Red blood cells are made in the red bone marrow, and then they're destroyed eventually. Red blood cells don't divide to make new red blood cells. Red blood cells are made in the red bone marrow through the specific process within hematopoiesis that we saw earlier. There is a homeostatic mechanism to regulate this, which is depicted here. This is from an old textbook. This is the way they depict homeostatic mechanisms. But basically what they're saying is when oxygen delivery to the kidneys primarily is low, the kidneys react to that to increase oxygen delivery by releasing erythropoietin, which is abbreviated EPO. We saw that last time. And that stimulates proerythroblasts in the red bone marrow to become reticulocytes that then become red blood cells. And so red blood cell numbers increase in circulation, which means that your blood's capable of carrying more oxygen. And so that increased oxygen carrying capacity will mean that the tissue is going to receive more oxygen. Okay. So that's the homeostatic mechanism that basically gets new red blood cells into the circulation as they're needed. The rate at which red blood cells are cleared out is pretty high. It's like a couple million, must be every day. I don't think it's every hour, but it's a lot of cells. I mean, there's 5 million red blood cells in 1 microliter of blood. You have, on average, 5 liters of blood. Okay, yeah.
If you only have 1 kidney, would that affect in any way the number of red blood cells you have in circulation?
The kidneys aren't making the red blood cells. Kidneys are just making the hormone stimulating red blood cell production. So 1 kidney, 2 kidneys, they're both going to release the hormone.
Okay.
So, but yeah, so yeah, you got 5 liters of blood. 1 microliter of blood is 5 million red blood cells. A microliter is a millionth of a liter, if I remember correctly, which means that you have 25 million red blood cells in your body, okay, on average. So the turnover is going to be in the millions, probably a couple of millions a day or something like that. But that's all regulated here. There's sort of this ongoing erythropoiesis process going on, depicted in this homeostatic mechanism. So would have been kind of nice to wrap up the red blood cell stuff at the end of the first part, but time just wasn't on their side. It never is. I never get to that last thing there on time. So now I want to look at the other cells. With white blood cells, I just want to introduce them and let you know that they exist and talk a little bit about them, but we're really going to look at them more in the immune system chapter, where their function is primarily discussed in the book. Leukocytes, or white blood cells, leuko means white, come in 5 different types. 3 of them, you see their names end in phil, neutrophil, eosinophil, basophil, and 2 of them, their names end in cyte, monocyte and lymphocyte. The names there actually separate them into the 2 main groups as we describe them. The neutrophils, eosinophils, and basophils are referred to as granular leukocytes, and the monocytes and lymphocytes are called agranular leukocytes. Granular refers to the fact that in the cytoplasm of the cell, we may or may not see grainy inclusions. That's what granular means. To my eyes, I suppose, the cytoplasm of the neutrophil doesn't really look that different from the cytoplasm of the monocyte, but they are classified differently. That's just where the names kind of fall into the group. Neutrophil, basophil, eosinophil is referring to how they react to staining, which has to do with the pH of the stain. Neutrophil is referring to a neutral pH. Eosinophil is referring to an acidic pH, and basophil is referring to a basic pH. Has something to do with how they're stained. Has nothing to do with the cell in your body. It's the name just comes from how they're viewed under the microscope. But really, those 3 and the monocytes are more closely related to each other than the lymphocyte is to any of these. The main reason there is the lymphocytes are the only cells that are made from the lymphoid stem cell in the red bone marrow, and everything else is made from the myeloid stem cell. All of the granular leukocytes, the monocytes, are separate from the lymphocyte. I kind of repeated myself there, sorry. Now, you'll notice that for those 4 types of cells, they have very unusual looking nuclei. They are not just simple spherical looking nuclei. They're kind of broken up into little globules of the nuclei. They are one continuous nucleus, so there's always going to be a little bit of a connection from one globule to the next, which is not hard to see in the neutrophil or the eosinophil here. Over in the basophil, because of how it stains, its cytoplasm and its nucleus are kind of hard to distinguish apart from each other, especially in the projected thing there. But there is a little wisp of a connection from one lobule here to the other lobule here. It exists. They're together. And I'll show you a picture in a little bit that explains why they have funny shaped nuclei. Lymphocytes do not. Lymphocytes have nice roundish nuclei. They're spherical in shape. While I'm still on this frame here, I want to mention down at the bottom it says, "Never let monkeys eat bananas." That's a mnemonic to help you remember about these cells. The N in never corresponds to the N in neutrophil. The L in let corresponds to the L in lymphocyte. The M in monkeys to the M in monocytes. E in eat, E in eosinophil. B in bananas, B in basophil. It obviously isn't in the same order as they're presented in this picture, partly because it reads better in that order, but also, more importantly, this tells you how many, or how prevalent each cell type is. The most prevalent type of white blood cell in your body is a neutrophil. It makes up about 2/3 of the cells, white blood cells, in your system. Lymphocytes are next. They're about 1/4, 20 to 25%. Monocytes are next. They're somewhere in the range of 8 to 10%. Okay, 10 or a little less percent. And then eosinophils and basophils are the least common. Collectively, they're going to be a few percent. Eosinophils are more common than basophils, and that's why they're in that order, but they're both quite rare. And again, we'll talk about the function of these cells more when we get to the immune system chapter, where their function is more crucial. Now, the reason why they have these odd-shaped nuclei is basically because of what's shown here in the picture. White blood cells tend to be found around the walls of the blood vessels. They're kind of rolling along the walls for the most part as they're going through circulation. And they can connect to molecules in the wall of the blood vessels, and they'll stop moving. The reason this happens is because the white blood cells are needed outside of circulation in tissue that's been exposed to something causing damage or disease. So cell surface molecules, represented by the green squares, connect to molecules on the inside surface of the blood vessels, represented by the things that are kind of like springs there. And it stops them from rolling along, and they stick to the blood vessel wall right there. Those spring-shaped things, which it says down at the bottom, are called selectins, are expressed by blood vessel cells when there's damage to the surrounding tissue. And so it stops the white blood cells in that particular neighborhood. And once they're stopped, they're going to squeeze out of the blood vessel and into the surrounding tissue, working their way between the gaps in the epithelium lining the blood vessel. It's a lot easier to squeeze through there if you have a nucleus that's all sort of separated out into little globules, kind of like link sausages that are all connected together. To squeeze a big spherical nucleus through a hole like that is a lot tougher. So the nuclei allow them to kind of squeeze through there, which is needed when they're called on to help fight off infections or help repair tissue or whatever it might be. And again, we'll talk about that a bit more when we get to the immune system chapter, but this is why they look like they do. Now, the next blood type to talk about would be, the next cell type to talk about would be the platelets, but instead of talking about them specifically as cells, we really want to talk about them within the context of blood clotting. Now, the term that means blood clotting is hemostasis. It's possible that you might see hematostasis somewhere, but usually hemostasis is the version that you see. Don't confuse that with homeostasis. Homeostasis, homeo means self. Hemostasis, hemo means blood. And it's how we stop blood loss. There are 3 major things that happen in this, which are in this little bullet list down here. Vascular spasm, platelet plug, and coagulation. In the book, and I'm going to go to the page in the book in a little bit to kind of point this out to you, it says the exact same thing. This particular presentation I've had for years, and it's using pictures from another book, but our current book covers it exactly the same way. Now, a vascular spasm is where the smooth muscle in the wall of the blood vessel contracts. It's a reflexive reaction to blood loss or damage to the blood vessel wall. In doing that, it narrows the blood vessel so that the blood flow is restricted. Okay? Blood flow and blood vessel diameter is really just a plumbing issue. Okay? You can move more water through a wider pipe than through a narrow pipe. Water will flow faster through a wide pipe than a narrow pipe. So by narrowing down the blood vessel diameter, it limits the amount of blood flow through that particular damaged blood vessel, which is going to limit the amount of blood loss from that. And there's a picture here that shows this, and the picture in the book is actually really good about that. The platelets then are responsible for temporarily stopping blood loss by plugging up the hole in the blood vessel wall. Platelets are also known as thrombocytes. Thrombus means blood clot. So these are the cells that are responsible for blood clotting. The actual sort of final blood clot isn't platelets necessarily, but rather it's made up of some protein structures that we'll talk about in a second. But as the protein structure forms to become that final blood clot, it traps the platelets in it. It also traps red blood cells in it. Not that red blood cells have anything to do with the clotting process, but there are so many of them that they're going to get caught in there too. And so clots are usually reddish in color because they have red blood cells trapped in them. And then the last thing is coagulation, which is a word that just basically means blood clot. And this is the series of events that creates the molecular structure that's a blood clot. And the molecule that makes up that blood clot is called fibrin. It's a fibrous type of protein, and it forms kind of a mesh, which is the clot. When this happens on the surface of your skin, when you cut your skin, you're bleeding out there, the blood clot that forms is what we call a scab. And that's kind of what we're talking about here. Now, the vascular spasm essentially is when the blood vessel is injured, and this is a rather gruesome picture of it, that they're like hacking through it with a razor blade or whatever. But whatever might happen to damage the blood vessel is going to immediately, reflexively cause the spasm, the narrowing of the blood vessel, because all of the smooth muscle is going to contract. And by narrowing down the blood vessel, it limits how much blood can go through and therefore how much is going to be lost. Then the platelets are going to form a plug sealing up that break in the wall of the blood vessel. This is stimulated by a number of different chemicals. We're not going to talk about what all is involved. You will see across a lot of what happens here that calcium is very important for blood clotting. But specifically what these chemicals are, we're not going to get bogged down with right here. What happens is platelets are just floating around in the blood, so to speak, or flowing around in the blood. And then something happens to activate them. And platelet activation is a feed-forward mechanism or a positive feedback mechanism. Those two terms mean the same thing. What that means in this case is when a platelet is activated, so one of these chemicals activates a platelet, it then releases its own signals, which would fall into the platelet factors group there. And so an activated platelet releases chemicals that activate platelets, and then newly activated platelets release chemicals that activate platelets, and more activated platelets activate more activated platelets, and activated platelets activate more activated platelets. It's positive feedback. It just suddenly blows up, and all of this happens really quickly. The point is, the faster that you have this response, the faster that you plug up the hole and the less blood is lost in the process. With any positive feedback cycle, the cycle's shut down by something else. So the platelets will just go on activating each other as long as they can, but something else shuts everything down. And it's essentially when blood loss is stopped, the platelets stop being activated. Now this is happening kind of immediately to plug up the hole. But also, while this is happening, other things are sort of happening at the same time. So this picturing is happening kind of alongside platelet plug formation, but there's more factors involved and it takes a little bit longer for it to happen. This is the coagulation phase, and this is a series of molecular chemical reactions within the blood that lead to the formation of the fibrin mesh that's going to be the blood clot. Now, it's actually separated into 3 different pathways. 2 separate pathways converge onto the third single common pathway. And those 2 separate pathways are called the extrinsic pathway and the intrinsic pathway. Extrinsic means outside of, intrinsic means within. So the extrinsic pathway are those things that have to do with stuff outside of the blood that causes blood clotting. And the intrinsic pathway is things within the blood that cause blood clotting. Now, the book is a little bit fuzzy on that point. I don't really like the way that our current book presents it. So just understand it's a matter of what's in the blood versus what's not in the blood. And not in the blood includes the blood vessel wall. That's not the blood, that's a completely different tissue. When tissue is damaged, any tissue is damaged, it releases some chemical signals saying basically, hey, I'm hurt, somebody help me. And the tissue damage releases this tissue factor stuff, which will then lead on to various other changes in the intrinsic pathway. And that is about all I want to say as far as the detail there. This picture doesn't go into a lot of detail on its own. The book does, and I'm going to show you that picture in a little bit. Mostly to say, don't get bogged down on the detail, just understand the basics behind it, which is what I'm talking about here. The damaged tissue releases chemical factors that initiate this process. On the other side, the intrinsic pathway, this is about what's happening in the blood, which is really about what the platelets do. Now, where the book kind of gets a little bit confusing on this, it's somewhat understandable why it does, but it's really about the intrinsic pathway is really about what's happening in the blood, components of the blood. Platelets are activated by signals from damaged tissue, okay? Or they're activated simply by being exposed to collagen. Now, there's no collagen in the blood, but once the blood vessel wall is ruptured, connective tissue below the epithelial lining, so these down here, this suggests loose connective tissue. There's collagen there, and that collagen is going to therefore be exposed to the platelets. And platelets, when they see collagen, become activated. Okay. And so that starts the intrinsic pathway. And various things happen here going through this pathway. And then whether it's the extrinsic or the intrinsic pathway, they both converge on this thing which is called factor 10, not factor X, factor 10. It's a Roman numeral, which then stimulates the activity or production of what's called Prothrombin Aids. The ASE ending on that word tells you that it's an enzyme. And what Prothrombin Aids does is it converts prothrombin into thrombin. Now, when we see something like this, it starts with pro, so there's prothrombin and thrombin. The one that has the pro prefix on it is what we call a proenzyme. or a precursor to an enzyme. Enzymes have an active site on them, and that active site is where the chemical reaction takes place. Proenzymes have something blocking the active site, and they get activated when that something blocking the active site is removed. It's usually a little bit of the end of the protein that's folded over blocking the active site. So prothrombin A's goes in and separates out that little bit of the protein. If we look at the words here, let's pretend the letters in the word prothrombin are the amino acids that make up that protein. And so what prothrombin A's does is it cuts between the O and the T, so that it removes part of the prothrombin molecule. And what's left over is thrombin, okay, or the now activated enzyme thrombin. And what thrombin does is it converts fibrinogen into fibrin. Now, fibrinogen is the third protein, plasma protein that I introduced last time. Remember, there was albumin and globulins, and then fibrinogen is the third one. There's a lot of it in your blood, and its job is to wait around until thrombin converts it into fibrin. Fibrinogen is a soluble protein. It dissolves into the plasma, and it floats around. Fibrin, which is basically a fragment of the fibrinogen molecule, is insoluble, meaning that it doesn't dissolve into the plasma. And so when fibrinogen is converted into fibrin, which happens at the site of blood clot formation, that fibrin molecule no longer dissolves into the plasma and it falls out of solution. So these little lines here suggest fibrin kind of falling out of the plasma into where the clot needs to form. Okay. What's not depicted here, actually a few things not depicted here, are other things that thrombin does. So thrombin primarily is an enzyme that breaks fibrinogen down so that we have fibrin to form the mesh of the clot. It helps to make that mesh. So besides just breaking fibrinogen down into fibrin and the other stuff, it also takes fibrin and forms the cross links in the mesh to hold it together. And that's going to stabilize the clot. Now, another thing that's left out of here for thrombin is that it's also involved in a positive feedback cycle, actually two positive feedback cycles. One of them is very short. Thrombin, when it's activated, because prothrombin A's has cut down prothrombin into thrombin, while it's going to go on and do the stuff I just talked about, it will also feed back and activate more prothrombin A's. With more prothrombin A's, you can convert more prothrombin into thrombin. And then you have more thrombin to feed back and activate more prothrombin A's, which is going to convert more prothrombin into thrombin. And thrombin is going to be available to feed back and activate more prothrombin A's. It's a positive feedback loop there. Thrombin will also feed back a little bit further into the intrinsic pathway, kind of around the activated platelets stage, and increase activity in that pathway, which will eventually get to factor 10, which is going to activate more prothrombin A's, which is going to convert prothrombin into thrombin. And so there's thrombin available to go into the intrinsic pathway, increase that activity, which is eventually going to activate more prothrombin A's, which is going to convert more prothrombin into thrombin, and that thrombin can go ahead and do the whole thing all over again. Okay. Again, positive feedback cycles make for a faster, quicker, whatever you want to say, more robust signal. And whether it's the platelets activating more platelets or thrombin either directly or indirectly activating more prothrombin A's, the point is to get to a very fast, stable clot that'll stop blood loss. And so we see that a lot in blood clotting. Now, the problem with positive feedback cycles like this is that they can get out of control. Okay? Something outside of this shuts it down, which is essentially the when blood loss is limited, it's stopped. But sometimes you can start a positive feedback cycle when there's not actually blood loss, and that can cause big problems. if you've ever seen a Plavix commercial, they're kind of talking about this. So Plavix is a drug that helps to prevent blood clotting in inappropriate times. And it's mostly given to people that have atherosclerosis or cholesterol plaques in their blood vessels, which will narrow the blood vessel a little bit. And when blood vessels are narrow, platelets run into each other. And when one platelet runs into another platelet, they activate each other, okay, which is part of the intrinsic pathway. So the intrinsic pathway doesn't have to be started by exposure to collagen or something like that. But anything that activates a platelet will get it started. And a narrow blood vessel because of a cholesterol plaque or something like that will do that. Plavix, as a drug, acts against that. It keeps the intrinsic pathway from being activated, which happens more often in people with cholesterol problems or something like that. So the point there that the intrinsic pathway, which is part of that thrombin positive feedback, or platelet activation, which is can potentially get you into problems and create blood clots when you don't want them, when you're not actually losing blood. And so a drug like Plavix prevents that particular part. It doesn't prevent blood clotting itself. It just prevents some of the positive feedback cycles that can cause problems. But if you cut your blood vessel, then the extrinsic pathway is going to be right there to do its job, and you're going to stop blood loss just the same.
So does each clotting factor do its own thing?
Yeah, basically. So, and I'll have a little bit more to say about that in a second, or a little bit less to say about that in a second, and you'll understand what I mean. But so for the series of pictures I'm going through here, oh, come on. Computer's a little slow. It should move to the next thing right in a second, which is not actually a picture, but rather a sort of a final statement, which is when the blood clot forms, whether it's the platelet plug or the fibrin mesh, in both situations, the platelet plug or the fibrin mesh basically attach to either side of the rupture in the blood vessel, and they contract a little bit. They pull the cut ends together, and that helps for the tissue there to heal. Come on, you're supposed to move. Okay, so you can see up in the corner there, it has a number 4. Okay, somebody on campus is playing games on the network, and so it's really slowing things down. No, probably not. It's this computer. But
if you hit escape, will it get big?
yeah, probably if I hit escape, it'll go out of this thing. Let's see. Yeah, there we go. So anyways, what I just said is this little blurb right here next to the number 4, if you want to. But more importantly, I want to get to this text field here. I'm not going to leave this up on the screen long enough for you to write everything down. You can get to it yourself. All of this is being saved as a YouTube video that you can go back to and you look at if you want to. But this is just hit some of the high points that are important in understanding the clotting thing. Some definitions of the extrinsic and intrinsic pathways. One important thing is the extrinsic pathway reacts very quickly because that's only going to be active when you are, in fact, losing blood, when a blood vessel has been damaged. The intrinsic pathway is a little bit slower because sometimes it might be activated by accident. And it allows a little bit of time to be shut down when it's accidentally activated or something like that. The common pathway ends in, is where both of those end. And it's about prothrombin Aids being formed and prothrombin converting to thrombin and fibrin threads being made, the insoluble protein, and thrombin helping to stabilize that. And then at the end here, what wasn't in the picture I just showed you and is not in the picture from the book, the positive feedback cycles that thrombin is involved in directly. One is activating more prothrombin Aids directly, and then also increasing intrinsic pathway activity, which is really about activating more platelets. Activated platelets have their own sort of thing, which is a positive feedback cycle, but thrombin plays into its own version of that a little bit too. I did want to, where do we go? Go to the book real quick. Should have opened this window up already. And just show you what they have picture-wise for the blood clotting stuff, because I want to just point out a few things that I want you to concentrate on, or more importantly, not concentrate on. So let me go to this section of the book real quick. So basically, in the book, first off, they mention those same three things that I said before: vascular spasm, formation of platelet blood coagulation. And then they go on to have this figure, which is very similar to the figure we just looked at in what it says, but of course it's a different figure. And I don't really feel like waiting for it. So I launched it so we could see it in a little bit bigger than this, but essentially, the figure is set up so on one side we have images depicting the damage to the blood vessel and the vascular spasm. And then down here, this would be where the platelets start to form a plug, and then down here finally, or maybe it's off the end here. Oh, maybe it loaded here finally. Yeah. And then finally, the formation of the actual fibrin mesh blood clot there. Alongside this, it has these cascades written in. So this gets at what the clotting factors are. They're numbered by Roman numerals. I pointed out earlier factor 10, because that's sort of the beginning of the common pathway. I really don't need you to worry too much about what these numbers are. As far as, these here, which are what, 12 and 11 and 9, 8, 7, 3. Don't worry about what those numbers mean so much. I'm not going to ask you any specifics about what's happening in the intrinsic or extrinsic pathway. Down here, I pointed out factor 10, which is mentioned here, and this is where we start the common pathway, which is what the other two pathways converge on. I think this green thing right here, which is factor 5, I think that's Prothrombin Aids, which in the picture I was showing you before, they provide the name, which is what I care about. Don't worry about the fact that it's called factor 5, if in fact that's what it is, but rather just prothrombin Aids is what converts prothrombin into thrombin. And then prothrombin, thrombin, fibrinogen, fibrin, et cetera, they have numbers associated with them too. Don't worry about the numbers, just the names that we've gone over. I think this one up here, that's the first one that they show for the extrinsic pathway, that's the tissue factor that's released by the damaged tissue. Okay, and I'm mentioning that because in the picture here they have intrinsic pathway, and they say damaged vessel wall, and then this extrinsic pathway, and they say trauma to extravascular cells. That's not exactly what intrinsic and extrinsic means. The intrinsic pathway can, in fact, be started by damage to the vessel wall, but what it is the collagen that's exposed by the damaged tissue then activates the platelets. But other things can activate platelets too, and that's what the intrinsic pathway is. The extrinsic pathway is trauma to the, not extravascular cells, but rather the, I don't know if this is exactly how you'd say it, the extra hemolytic cells? No, anyways, cells that are not in the blood. Okay, that word there, extravascular, says cells that are not the blood vessel. But in fact, the blood vessel by itself is often the start of the extrinsic pathway. Okay. And so that factor 3 there is what I was showing you in the previous picture. That's the chemical released by damaged tissue, which then starts the extrinsic pathway. So really just pay attention to what is starting these. It's what activates platelets versus signals coming from damaged tissue. They eventually both converge here, which activates Prothrombin Aids, and then we have all these things happening. Also in this picture, just like the picture I was showing you before, they don't stress the other things that thrombin does. Now they do say cross-linked fibrin clot down at the bottom. They don't mention that cross-linking is partly due to the actions of thrombin itself. And they definitely don't show any positive feedback to the Prothrombin Aids or into the intrinsic pathway. Whoops, went the wrong way. Intrinsic pathway over here. But in the text slide I just ended the presentation with a second ago, that's written out, so you see that there. Okay. And the books don't always highlight those positive feedback cycles, but I think it's important because, again, positive feedback is crucial in blood clotting because it quickly stops blood loss. That's the point of blood clotting. It's to limit how much blood is lost from the circulatory system. So, now if we were to go back to the presentation, and I'm not really going to, but it goes on to, whoops, just say something about blood typing. This is something that I handle in lab, so we're not going to talk about it here. The book also has a section on blood typing, which we won't go through here, but just to know that it exists and they deal with it there. So that kind of wraps up the blood stuff. Okay. Now, in the schedule for the course for today, it says blood clotting / intro to heart. Since I still have 15 minutes, I would like to just introduce the heart a little bit, get us started there. The heart lecture material is actually going to be rather involved. We kind of need more than just a week's worth of time, so I want to start it now. So we're at least a little bit further through when we get to starting that next week. So again, I'm going to be using presentations. Over the weekend, I'm going to actually put pictures from the book into this. I haven't done it yet, but I'm going to show you some pictures today, which come from old books, but when we continue this on next week, I'll have pictures from our current book in there. Okay. So what I really want to talk about today is some of the anatomy about the heart, where it's located, and a little bit about what makes it up. You're probably familiar with this already, but the heart is in the thoracic cavity. It's located between the lungs. The region where it's located is actually referred to as the mediastinum, which is the region in the thoracic cavity that is between the lungs. The upper limit to the mediastinum varies depending on the source that you're looking at. And I don't really care exactly where we call the top of the mediastinum. If it's right here, right above all the structures of the heart, or if it goes up into the base of the throat, whatever, that's not terribly important for understanding the heart. But the bottom of the mediastinum is very definitive, which is the diaphragm. Okay? So the heart basically sits right on top of the diaphragm, and then it sits between the lungs. Other things are found in the mediastinum besides the heart. Okay? The esophagus, which is going from the oral cavity to the stomach in the abdominal cavity, passes posterior to the heart. And going from the head and neck down to the lungs is the trachea. The trachea is located in between the esophagus and the heart. Okay, so as we're looking at this anterior view, we can see the trachea exposed here. The esophagus would be just posterior to that, which we can't see from this point of view. But both of them pass behind, posterior to the heart. The trachea ends near where the heart is, splits into the bronchi and which go into either lung. The esophagus continues on and actually passes through the diaphragm there. One of the structures we're going to talk about in understanding about the heart and circulation is the aorta, which is the artery that comes out of the heart. And we see that in this picture, this big red arch right here is the aorta. It obviously comes out of the heart, which is what we're talking about here. It arcs over and then descends posterior to the heart, but it actually descends posterior to the esophagus. So right now we're looking at the heart, the trachea we can see, which is just behind the heart, and then the esophagus, which we can't see, would be just behind that. Those both pass next to the aortic arch, and the aortic arch goes all the way past them and descends in a very posterior position, basically right next to the spinal column. Those are some of the major players that we find in the mediastinum. In this transverse section through the thoracic cavity, we can see some of this. So the heart here at the bottom of the picture, which is anterior, we see the aortic arch coming up. They've cut the aortic arch off, and it continues over here next to the vertebral bone. And at the level that we're cutting through, the trachea is no longer present. We can see the two bronchi on either side, but they would have emerged from the trachea, which would have been adjacent to the posterior surface of the heart and separating that from the esophagus, which we can see right here in that position. Posterior to the heart, a little bit anterior and more medial than the descending aorta. In this picture, you can see that there's some empty spaces around the heart and around the lungs. When we get to the lungs, we'll talk about what that is, but around the heart, it's surrounded by a serous membrane, which helps to kind of separate it from the surrounding organs a little bit. And that serous membrane is referred to as the pericardium. Any serous membrane is a simple squamous epithelium and its associated areolar connective tissue. That's a membrane, serous membrane. For this one, it covers directly the outside of the heart, and we'll see actually it is the outermost layer of the heart itself. But it extends past the heart, and it then sort of folds over and lines the inside of the mediastinum right around the heart. The apical surface of the squamous cells are away from the heart for the part that's adjacent to the heart, and then away from the mediastinal wall for the part that's on the other side. The apical surface of those two layers are actually facing each other, and then there's a gap between them. That gap is filled with a fluid, what we would call a serous fluid, which is actually kind of a weird construction because the word serous comes from the word serum, which is a type of fluid. But we don't call that serum because serum is actually used in a different context in the body. But that serous fluid fills up that space between the inner and outer layers, what are called the parietal and visceral layers. The parietal layer is the one that's lining the inside of the mediastinum. Parietal essentially means body wall. And the visceral layer is the one is a side that's actually adjacent to the heart. Visceral referring to the organ of the heart. And between the two, there's this fluid that's accumulated. That fluid acts as a lubricant. As the heart's beating, the pericardium surrounds it, and it cuts down on the potential friction between the moving, beating heart and the things around it, like the lungs and the diaphragm and the esophagus and all that sort of thing. The parietal pericardium is actually a little bit more than just that simple squamous epithelium and its associated areolar connective tissue. There's also a rather thick layer of fibrous connective tissue, dense, irregular connective tissue, which is usually referred to as the fibrous pericardium, or here it's called the fibrous tissue of the pericardial sac. It surrounds the whole thing and kind of anchors it in place. And we can see here at the bottom of the picture that fibrous pericardium is actually anchoring it to the top of the diaphragm, holding everything in place. I've always kind of just gotten a kick out of this picture, but if you're trying to visualize what we're talking about here, imagine a balloon that is rather deflated, and you take your fist, which is about the size of the heart, and wrap that deflated balloon around it. The wall of the balloon represents that serous membrane. Some of it is going to be directly adjacent to your hand, and then there's going to be another wall that is opposed to that layer that's against your fist. So that's kind of the same idea. If you imagined this balloon were filled with a little bit of fluid, then that fluid would fill in that space between your hand and the outside wall there. But they call it an air space here because apparently the book can't imagine such a thing as a water balloon. It has to be an air balloon. But that's one way of kind of picturing what's going on here. Now, having described the pericardium, I want to go back to this picture for a second to point out a couple of other things. As we're looking at the heart, it has an apex and a base. The apex is kind of where it comes to a point at the bottom of the picture, sort of the inferior most point on the heart. And then the base, represented by this line, is sort of the top of the heart. Now, the words base and apex, when we're using them to describe a mountain, base is the bottom, apex is the top of a mountain, but apex is also the pointy end of the mountain. So the pointy end of the heart is the apex, and then the base is just opposite that. The base is where the heart's connected to everything else. So there are a bunch of major vessels, which we'll talk about in lab, that are where the base is. The heart, while we think of it as being kind of in the center of things, is a little bit to the left side, which I should actually go back. Oh, great. Things aren't moving yet, but we saw that picture earlier. It's a little bit off to the left side. There is, in fact, a gene which has the cute name of lefty, which is, for some reason, I don't know exactly how this works, but it's the signal that tells the heart to develop a little bit off center on the left. There are very, very rare people that have a mutation in that gene that doesn't work. And so their heart actually doesn't develop off to the left side. It develops off to the right side. Very rare condition. You're probably never going to meet somebody with that condition. However, apparently it's an interesting enough condition to make it onto an episode of CSI once. I think it was CSI.
My nephew has Insitus inversus.
So that might be it. Okay, so it's not as rare as I'm trying to.
It is very rare. He's like one of those anomalies that whenever he goes into the hospital, everybody wants to listen to his heart because it's forward.
Yeah. So CSI, I think it was, or whatever. Somebody was shot through the chest. What should have been a fatal shot because it would have pierced the heart. But the person survived, and they find out eventually the reason they survived is because their heart is on the other side of their chest. Again, this kind of weird position. So I wouldn't suggest to your nephew, is that what you said?
Mm-hmm.
That he go around getting shot in the left side of his chest, but still.
He goes up to doctors and he says, "Try and find my heart. Go ahead." It's fine.
So yeah, it's a very rare condition, but it is a genetic signal that tells the heart to develop a little bit onto the left side, and if there's an error in that gene, then it's not going to happen. So, but that's a fairly rare condition. Okay, so let's see if this is going to. Okay, I think this has just completely stopped moving, which is fine. The next thing that this would get into is the basic structure of the heart, which is part of what we're going to be covering in lab, or we did cover in lab yesterday for the Wednesday group. So we'll pick up with that. I don't think the Monday lab group will get to and then we'll have lecture on Tuesday where we'll continue on with the heart stuff. And then on Wednesday, the Monday lab group meets, not the Wednesday lab group, but the Monday lab group meets, and we'll do the heart lab thing. So I'm going to go over a few things after this in the presentation about this basic structure of the heart, which I'll repeat again in more detail on Wednesday for that half of the group, but that's where we're going. So everybody but Crystal and Natalia, you're welcome to leave. Yeah, you guys.
When do you have a meeting for, what time on Wednesday?
Same time, same place. Just Wednesday is a Monday.
Wednesday is the day.